We have two groups. Those you can grow in your laboratory, called heterotrophic bacteria, and those that don't grow, but you can stain them and see them as present by microscopic means, which are called VBNC bacteria. And the best way of seeing the bacteria is by the live dead technique. You can buy kits. It's a double staining procedure. One stain picks up what we call the live bacteria. These would be your heterotrophs. And the other stain, because of damaged membranes to the VBNC bacteria, are what we call dead, but they're not really dead. They can recover. And when they recover, they increase your microbial population. And there's other ways. Cell elongation. If you take seawater and you add antibiotics to it to prevent cell division, and then add nutrient, the VBNC bacteria elongate. They just don't divide, and you can really see them. They become about 1 micron in size. Very easy to see them. Very difficult when they're 0.1 to see them microscopically, because that's just about the at the limit of resolution for a regular light microscope. I won't go into this. Okay. Now the thing is, research demands, how do you transform VBNC back to the normal heterotrophic form? And it's the normal heterotrophic forms that form the biofilm. And that triggers resuscitation. What triggers resuscitation? That's an important area of research. And here in the laboratory, you can, by certain means, and one of the best ways is take a 30-degree organism from seawater and try to grow it or induce it to go to like 5 degrees C. The organism, the temperature differential then becomes a VBNC bacteria, and you see that the number of viable cells that are plate counts goes from 10 to the 6 to 10 to the 1. So the bacteria here are in this VBNC state. There are other ways to induce VBNC. Low nutrients do it. Certain heavy metals do it. And here you can see other bacteria you can induce into the VBNC state, and what happens is the heterotrophic counts really decrease. But you can also take VBNC and resuscitate them. You see here, you go from 10 to the 1 up to 10 to the 6 by a resuscitation, and it could be a temperature differential. It could be also certain triggering molecules hitting certain receptor sites on the VBNC. Now all of a sudden, you go from 10 to the 1 to 10 to the 6. There is no growth taking place. You get a tremendous increase in your heterotrophic population, but it's all due to VBNC resuscitation. And you think growth has taken place, but it really hasn't taken place. It's just a resuscitation of the VBNC. Now, this was from a study I did in Tampa, Florida, on the heterotrophic, and DAPI is a stain that picks up all the bacteria. And you see at Tampa, the heterotrophic counts, you see the decrease from 10 to the 4 to 10 to the 1 because of chlorination. They were doing chlorination, dechlorination. But you see, at the point of dechlorination, you went from 10 to the 1 to 10 to the 4, eventually to 10 to the 5th. But the actual total number of bacteria, the VBNC, were 99.9% of the population. So here we got resuscitation. What we saw here is resuscitation, not bacterial growth taking place at Tampa. And if you looked, in my career, I looked at various sites in the Middle East, at Kuwait, Kisser, Alberg, Jeddah, Ad-Dur, and Tampa. And the only place that we didn't see biofouling taking place was in Jeddah. Okay. Now all these other sites were doing chlorine dechlorination. Jeddah, they went to intermittent chlorination. They would chlorinate at the intake for 8 hours every week. And that was it. And you see that nowhere in Jeddah did they get any type of resuscitation taking place. Whereas in all the sites in the Middle East and in Tampa, resuscitation had taken place. If you look at the growth cycle of a bacterium, you can see that normal growth, logarithmic growth, stationary, and then death. But the thing is that you can, this is growth, and if you follow the Monod equation, you can measure the growth rate because there's a certain products being produced by the metabolic activity. But if this is resuscitation, you go from this point to this point without any significant growth rate. Okay? All it is bacteria. So you can get to high numbers in seawater without bacterial growth taking place, just by resuscitation. And that's the way I think that the huge numbers of bacteria entering the RO membrane are achieved is by resuscitation. Now, the organics are the other component, and of course, Loren showed you that you can measure these TEP, very simple procedure, 0.4, 0.1, and you can actually use a 0.05 micron. We're interested in this 0.1, 0.05. The 0.4 are the large particulate. So in our studies for these glycoproteins, we're interested is what passes through the 0.4 but retained on the 0.1 or 0.05, and you can measure these concentrations at 787 nanometers. And Loren showed you that the way I think it takes place, and Loren probably is correct, is that the algae in the seawater are the source of not only TEP, but is the source of these glycoproteins that also stain with this Alcium blue. Only thing, there are very low molecular weight compounds, not these large TEP molecules that you can envision that are retained by 0.45 micron. Now, one of the things, experiments we did is if you grow algae in the laboratory, the algae are going to be producing various compounds, various glycoproteins. And with different organisms, at a certain point, if you take the exudate from the algae and put it in with the bacteria, so here's one organism that was now going into the VBNC, here's another organism going to VBNC, all because of the exudate from these algae. Okay, so here, what's being produced by the algae are inducing the VBNC population. But then at a certain point, there are substances produced by the algae that are now resuscitating the VBNC. So, and this is an area of research that we're doing, is we think there are various substances produced by the algae that actually help induce VBNC, and then there are substances that take the VBNC back into the heterotrophic form that can bruise these EPS material and produce the biofilm on the membrane surface. So we're looking at isolating these different type of glycoproteins produced by the algae. Maria Kennedy, and you can use this technique of LCOCD. If you look at seawater, you can see the various groups of organics involved that are present in seawater. It's a very neat technique, LCOCD, and we're trying to identify, once we get these glycoproteins, where they exist. Now, one of the things about these materials produced by the algae is they have lectin-like ability, which means that they can agglutinate cells, they enhance bacterial attachment, very important, they interact through the carbohydrates that are present, and you can use an assay with a algae called Chlorella vulgaris to measure. So one of the ways that you can measure whether a seawater sample has these glycoproteins that aid in the adhesion of bacteria is by doing this Chlorella assay. And here you can see, let's go back. Here you can see that the control, the chlorella, do not agglutinate with each other, but if seawater has these lectin-like glycoprotein, the chlorella starts to clump together.
5 minutes, Harvey.
Okay, so let me get to importance of flux, concentration, polarization, and critical flux. We all know about. Let me get to how to control biofilm formation by controlling flux. Now, in 5 minutes, I'm going to tell you about a pilot plant we have in the Caribbean. It's a seawater. It's built by GE, flux of 8.3, cross flow velocity based upon a feed flow of 34 gallons per minute recovery. And this thing is, we studied a new technique called internal hybridization, where rather than having one type of membrane in the pressure vessel, we have a low permeable membrane up front, and in the back we load it with various high permeable membranes. Now, what happens is our pilot plant, you can see, had a decrease in flux, the control. So the biofouling was taking place with the normal array and the cross-flow velocity that we achieved with 34 gallons per minute. And you can see the energy consumption of this pilot plant with this low cross-flow velocity would increase. So what we decided to do is to increase the cross-flow velocity to see if we can control the fouling by minimizing or increasing the cross-flow velocity. And what we did is the high pressure pump was not sufficient to increase the feed flow, so we just reduced the number of pressure vessels to increase the system flux to maintain productivity, and we also eventually went to this hybridization. And you can see by doing 10 pressure vessels versus 14, you have a much greater cross-flow velocity than in the control. The problem is when we went to this 10 to 7, we used these very high permeable membranes. And it turned out, even though we had a greater cross-flow velocity, our fluxes were so much higher that the same concentration polarization effect occurred, and we couldn't control fouling even by increasing cross-flow velocity. And here you see that the permeate flow rate would go down in both cases because we achieved a higher cross-flow velocity, but we had upped our flux. So the concentration polarization was the same for both cases. So now we went to the, and of course the energy use went up. So we went to the hybridization technique, and what happens is you can see we now have a greater cross-flow velocity. The system flux goes from 8.3 to 10.8, and we can control the flux on the lead end element where all the biofouling is taking place because we have low permeable membrane, and all of our high flux are in the brine end element, so that there is very little biofouling taking place, and that's just hydraulic evaluation. But the thing is that you can control the biofouling, okay, with the ISD. It's called ISD, Internal Stage Design. Various companies do this. Dow, Hydronautics do it. They do it in terms of for energy purposes because using these high permeable membranes. But it turns out it works in terms of control of biofouling because the low flux and high cross-flow velocities on the lead end element control the biofouling. Okay, so here you see the ISD design. The energy stays constant, but in the with the high flux,